And perfect. Okay, thank you, Daniel. Thank you all for joining us today for El Zoominario. Today, we're very excited to have Natalia Rosario-Melendez. Natalia is from Carolina, Puerto Rico. She did her bachelor's in cellular and molecular biology at the University of Puerto Rico, Rio Piedras Campus. She then came here to UW-Madison to do a master's degree in bacteriology with Dr. Mark Mandel, where she studied how bacteria transition from living freely to forming biofilms to colonize hosts. Currently, she's a third-year PhD candidate in Dr. Joe Handelman's lab, studying how different species of bacteria interact. Thank you very much, Natalia, for accepting this invitation, and please just get started.
Thank you, Claudia. Yeah, so today I'm gonna talk to you about bacterial languages, so how a multi-species microbial community communicates. Here. Okay, so microbes can be found in every environment in the world, and they can cover almost every surface on Earth. So I like to think that we live in their world. So we live in a microbial world, and usually when we think about microbes, most people think about pathogens and things that makes us sick, but most microbes hold a variety of interactions with their neighboring environments and microbes, from beneficial to pathogenic. So different species of microbes sharing the same space in an environment are considered a microbial community. And these communities are essential in all environments on Earth. They go from doing biogeochemical cycles, so nitrogen cycle, carbon cycling, to aiding digestions in humans. They're also our first skin barrier, and to affecting plant health. And for my talk, I'm going to be talking about microbes that affect plant health, a very specific niche in soil called the rhizosphere. And what is the rhizosphere? So the rhizosphere is the area adjacent to plant roots. And this area is very nutrient dense due to secretions from plant roots. And for this reason, this area in the soil has a higher population of microbes that are more active and has a lot of bioactivity when comparing it to bulk soil without any roots. So the rhizosphere is a very complex ecosystem where microbes can interact with their environment as well as other microbes in that same environment. So in order for us to be able to enhance plant health, we need to be able to understand and manipulate these communities and their functions in their environments. So to do this, we need to understand how the information flows through a community and how these communities are communicating with each other. So as I already mentioned, microbes are beneficial for disease prevention in plants, and this is one example in soy plants. So here in this red arrow shows a healthy plant and not just the control. And if then we expose the seeds of this plant to a pathogen, which in this case is a water mold, then we can see disease spreading, and like we have a sick plant, it doesn't germinate properly. But then in the third plant, these seeds were then exposed to a beneficial microbe and to the pathogen, and we can see that the plants are again healthy, and that the beneficial microbe was able to prevent disease in the plant. So the microbe that was inoculated here is called Bacillus cereus, and this is important later in my talk. But how the mechanism of how this happens is due to two small molecules that are produced by this bacteria, and they are called Zwittermycin and canosamine, and they have antibiotic activity against this water mold, and this is how the disease is prevented. So what are antibiotics? We might know some common ones like neomycin, bacitracin, and polymyxin B, and they are small compounds that are made by many organisms, but these ones right here are made from bacteria, and they are commonly found in ointments like Neosporin, triple antibiotic, and things like that that we might use on a daily basis. But being antibiotics is not their only function. These antibiotics are small molecules, and they might hold other big interactions because they are often, when they are produced in their natural environments, they are not produced at a concentration that they would often inhibit other microbes. So for this reason, it's been tested that small molecules mediate microbial interactions. So these small molecules are often not required for growth, and their synthesis, so how they are synthesized, is encoded in their genomes. So their genomes have these clusters of genes called biosynthetic gene clusters that transcribe the building blocks in order to synthesize these molecules. And then we also know that they can mediate cell-to-cell communication. However, this has not been yet studied in a full community and what the effects of these are. So my question, or the question we're trying to answer, is how are small molecules used as signals in microbial communities? And to answer this question, we first need a model or a microbial community, right? So for this reason, our lab developed the model microbial community Thor. It stands for the hitchhikers of the rhizosphere. And how it started was that Bacillus cereus, remember the one we talked about earlier that protects soybean plants from disease. So we were isolating Bacillus cereus from soybean roots. And this is just a plate of how the isolation process looks like. So each circle or colony represents a different bacteria that was in those roots. And then we further isolated the ones that were specifically Bacillus cereus. And upon the process of purifying it, we saw that there were other bacteria that were hitchhiking or physically associating with Bacillus cereus after a bit. So for this reason, we did some experiments and we looked at the interaction network of antagonism and commensalism of this bacteria. And we ended up choosing 3 bacteria: Bacillus cereus, Pseudomonas cariensis, and Phylobacterium johnsoniae, which are common bacteria that can be found in the rhizosphere in soil. And what's really also very interesting is that they represent 3 separate phyla, so three different groups of bacteria within the soil. And these bacteria have some really interesting behaviors when they're together. So we're going to talk about 3 of them today. And one of them is that whenever Pseudomonas cariensis is with Phylobacterium johnsoniae, it inhibits its growth. And how it inhibits this growth is through another small molecule. This small molecule is called cariensine. So since it's inhibiting, it has some antibiotic activity against Phylobacterium johnsoniae. However, whenever Bacillus cereus is there, in addition to Pseudomonas cariensis and Phylobacterium johnsoniae, then Bacillus cereus is able to promote Phylobacterium's growth and also protect it from the inhibition from Pseudomonas cariensis from this small molecule. So this interaction network suggests that there's some signaling and some communication that's being mediated by this small molecule. But how do we usually measure how species interact or communicate. And how we do this is usually, can be one of the ways that we can do this, I should say, is by looking at how the genes in those microbes are being expressed. So that's what we did. We looked at how the different genes were expressed, and we compared how they were expressed in a normal Pseudomonas coriensis that expresses, that makes coriancin, and a Pseudomonas coriensis that does not make this small molecule. And we compared the two, and we looked at the differences. And what I want you to get out of this graph, it looks scary, but it's not. So if there is a yellow bar, it means that that gene was expressed more in the presence of the small molecule coriancin. And if there's like a big blue bar, then it was downregulated. So its expression was shut off or diminished in the presence of in the absence of this small molecule. So here in the left, we have Flebobacterium johnsoniae, and in the right, we have Bacillus cereus. And the two things that I want you to get out of this graph is, one, that the small molecule coriancin can elicit a conserved response, so a similar response in two different bacteria, but it can also elicit some different responses in the bacteria. So there are things that are going to be conserved between the two, and there are some things that are going to be different. So in order to look a little more further on how genes are, how it affects the same genes that are conserved in multiple species, then I looked at one specific gene that, when looking at this data, we saw that was being expressed in all of them when the small molecule coriancin was there. So if we take away the small molecule, then we do not see expression at all. I'm only showing you here Flebobacterium johnsoniae, and the green bar means that the small molecule coriancin is present, and the bar represents that there was expression. And we don't have any orange bars because when that small molecule is not there, there was no expression. But this was something that we saw in the same gene in different bacteria. That same gene was in all of them. So this suggests to us that this gene is then important for all of the bacteria and how they respond to this specific molecule. So how is this interaction looking in the community? So if we have the small molecule coriancin, then that means that it signals to the other bacteria, but that it can also signal through to itself. And what were other things that that might be affected by this one small molecule was another, if you remember from when I spoke before, there were two small molecules that Bacillus cereus makes in order to protect plants, and those were pseudomycin and canosamine. There are also two small molecules. And here in this graph, you can see in the x-axis are all of the genes that are required to make those two molecules. Specifically in gray, or in part of this rectangle, enclosing this rectangle, are the ones that are necessary for kanamycin, one of the two molecules. And we can see that then this small molecule produced by coriancin, produced by Pseudomonas coriensis, is affecting other small molecules in other bacteria. So this brings us back to then like an interaction network of how the bacteria is communicating. So to wrap up a little and put everything into perspective again, then we have this bacteria, Pseudomonas coriensis, that produces this small molecule called coriancin. that elicits community-regulated genes in all of the bacteria in this community. But also we saw that it elicits it to produce other small molecules in Bacillus cereus that in turn will affect the communication in the roots. So with that, I would like to thank my lab and our funding sources. And for my culture talk, I wanted to talk about bomba y plena. And these are two folkloric music in Puerto Rico, and they have very big cultural and historic significance for us. They are often referred to like together and as the same thing, but they are not the same thing. In fact, they're very different. And bombas, the instrument used for bomba are the ones that are referenced with the red arrows, which are these three big barrels or drums, these sticks, and the maraca. And how bomba originated, it dates back to European colonialism, and it originated from enslaved Africans in the 17th century after Taínos, or the natives of Puerto Rico, were exploited and were dying, so they had to bring other labor. And to them, bomba was a source of political and spiritual expression, and its lyrics conveyed anger, sadness about their condition. And these songs were usually catalysts for rebellion and uprisings. And it is important to note that bomba is still used as an expression of rebellion against government in protests in Puerto Rico. It's something that it's very well alive and that we honor in our daily lives for the most part. And then plena started in the 20th century, and the instruments are in this square, yellow square. So it's 3 hand drums, or pleneras, which is what they're called, and a guiro, which I don't know if there's a name for it in English, but that's what the bass is. But it was highly influenced by bomba, but now it started in the 20th century when Jibaros, or Puerto Rican workers, and they usually use it we usually refer to it as the newspaper of the people, because at that time there were a lot of workers that did not know how to read or write. So a lot of messages were conveyed in song. So there are a lot of songs that are about like someone that got cut and went to the hospital, and like we can see that still today in some of the like older and traditional songs. Oh, and something really fascinating, and why it's one of my favorite dances about bomba, is that the dancer guides the lead drum. So there's a bass rhythm that is conveyed by all of the other drums, but there's one specific drum that it's the lead drum, and the drum follows the dancer. So I think this is something that's really unique. And to the dancer, we usually say habla, or talk, speak, and it's speaking through dancing, through her or their or his movements, which I think it's something really unique to this type of dance, and it's why it's my favorite. And with that, thank you for coming to my talk.
Thank you, Natalia. I think we can stop